Okay, so we left off talking about membrane permeability. And so remember that we have this phospholipid bilayer, right, as our plasma membrane. And when we look at that phospholipid bilayer, remember that we have this kind of nice, squishy, hydrophobic center, right? So if I can draw there we go. So we have this nice kind of squishy hydrophobic center right here that's with these fatty acid tails, that's nonpolar. And then here we have the polar heads right on the outside of the membrane here, okay? So they're charged and like to interact with other charged things. So just remember again that we have the hydrophobic center, okay? That's important to remember because remember that our plasma membrane is selectively permeable, which means that it allows some things to pass through and some things not so much. And so it's pretty easy to figure out what doesn't like to pass through because of this nice squishy hydrophobic center. So remember that just like oil and water don't mix, the reason that oil and water don't mix is that water is charged, right? So water is this charged molecule, and oil is not. But remember that water just has partial charges, okay? So meaning that you got a partial negative on the oxygen and partial positives on the hydrogens, okay? So what this means is this hydrophobic center doesn't allow things that are large, oops, I meant to write this up here, large and charged, okay? Kind of like that old saying, large and in charge, in those superhero movies. So, but here it's large and charged. So anything that is a really large, bulky molecule or anything that's really charged is not going to want to pass through this hydrophobic nonpolar center of the membrane, of that phospholipid membrane. So what this means though is that there are things that are large and charged that can't get through unless they have some help. Now, the membrane this is a little tricky, it is permeable to water. And I say that lightly because water will move through it, albeit slowly, unless you have like aquaporins or something in it. But water molecules can move through the membrane. And you may say, but they're charged. Well, remember that these are only partial charges. So they're not that charged, okay? So because water molecules are small and because they only have these partial charges, they can make their way through the membrane, albeit they can make their way through the membrane much more quickly if there are channels, protein channels that allow them through, okay? And we'll talk about that. So you can increase the permeability of a membrane to water because they're still charged, they'll move through the membrane, but they can move through the membrane much more rapidly if you have a protein channel. So keep that in mind. But it's important to know that water can move through the membrane because we're going to talk about osmosis in just a minute. So there are two ways in which substances can cross the membrane. One is the passive processes, meaning that there's no energy required. And we're going to talk about concentration gradients in a minute. And the molecules are just going to move right down their concentration gradients. And then the active process is going to be where energy, or in the form of ATP, right? Energy in the form of ATP is going to be required. And this is because we're actually going to pump things across their concentration gradients. Okay, so let's get into what this concentration gradient is and what we mean by that. So again, we're going to talk about passive membrane transport first. And this requires no energy, right? And there are two types of passive transport. There's what we call diffusion, and we can have either simple diffusion or sometimes we need some proteins to help that diffusion process. So we have carrier and channel mediated facilitated diffusion. I'll talk about this in just a minute because I know you're probably like, no, not this again, but this actually will be really helpful for you, especially in 202 if you kind of review this now. And then, of course, we have osmosis too. All of these diffusion, types of diffusion, simple diffusion, carrier, channel, mediated, facilitated diffusion, osmosis, they all do not require any energy, okay? So I have this nice little paramecium power liver cartoon here, right, that says, ooh, you want to pass through? Oh, sure, go right ahead. That's just fine with me, because again, these are things that are just passing through, like water molecules, like in osmosis, through passive transport that can actually make it through the membrane, okay? And then the other type of passive transport, we're not going to talk much about it now, but you will talk about it, especially more in Ch3 when you talk about blood vessels and you talk about the urinary system and what happens in the kidney tubules. So this second type of passive transport is called filtration. And this is a type of transport that usually, again, happens across capillary walls, okay? So where you're going to have substances, again, just going down their concentration gradient, especially water, across capillary walls, okay? So keep that in mind. Those are the main types of passive membrane transport. Again, we'll come back and talk about filtration more in Ch3, but we're really going to focus on diffusion, okay? So I always say you guys all intuitively know diffusion. Everybody knows what diffusion is, because if you were to put a skunk inside the classroom, right? And that skunk, or my dog is sitting behind me, so I always like to use that as an example too. If you have a dog and they break wind, I have a 150-pound Great Dane, and so when she breaks wind, right, it's quite a lot. She can do some damage because she's so large. And so what happens is all of those odoriferous particles, right, are all concentrated in that one area, right, when they first break wind, okay? Or in this case, if a skunk sprays you, or sprays in a room. And you can be sitting across the room, right? So my dog actually is in the office right now. I'm on the other side of the office from her. But if she breaks wind, I intuitively know that I eventually am going to smell those lovely odoriferous particles. And that's because they're going to move down their concentration gradient from that area where they were in a high concentration, right around her anus when she released her gas, to, again, me, where they're in a lower concentration, right? And that's because those molecules, those odoriferous particles, are going to move down their concentration gradient. What happens is they're constantly moving around, and the molecules, we think of air, the air above us as stagnant, but it's not at all. It's constantly moving. Those molecules are moving around. And so as all of those molecules are kind of moving around and colliding with each other, they're going to move from a high concentration down to low concentration, which, again, you all intuitively know. If your dog breaks wind on the other side of the room, you are likely going to try to open a door and allow the diffusion of those molecules into another room, right? So we all intuitively understand the concept of diffusion. So if you ever forget, just think of, those very simple things like your dog breaking wind. And you'll remember that things move from areas where they're in a high concentration down their concentration gradient to areas of a low concentration. And energy is not required for this to happen, okay? Now, the speed or rate of diffusion is influenced by things that we call, basically by Fick's law. And so, hold on, I'll go through that in just a minute. But here, this was just, I just put this up here because, again, you also all intuitively understand diffusion. If you were to put, again, a dye pellet, right, in a glass, in a beaker, you know that eventually the dye will be evenly distributed. So principles of diffusion, many of these are actually very intuitive as well. And you know a lot of these because you do some of these yourself. So according to Fick's law of diffusion, we have these principles by which there are these different factors that will affect the rates of diffusion. So obviously, the steeper the concentration gradient, the faster diffusion will occur, correct? Right? So your maybe small dog, okay, breaks wind, and then my 115-pound dog breaks wind. She probably is going to release a lot more odoriferous particles that are going to be in a greater concentration, right? So I may smell her lovely gas before I smell a small dog's gas, just because, again, her concentration gradient is going to be steeper. So the steepness of that gradient is going to affect the rate of diffusion. Temperature, again, you guys all intuitively understand this. You know that, right? Temperature increases the rates of collisions of those molecules because temperature just makes molecules excited, right? It makes them bounce around faster and faster. They get really excited, like I just probably did to my microphone here. And so we use these principles when we're doing something like making tea. Like we're going to go ahead and take our tea bag and we're going to put it in hot water because those molecules are going to diffuse much more rapidly. Our tea is going to diffuse much more rapidly into our hot water than it will into ice cold water. Okay, so obviously, the warmer the temperature, the faster the rate of diffusion. The size or mass of the diffusing substances. So again, substances that are really large are going to diffuse slower, especially through the membrane, right? If you're a large substance, you're going to diffuse much slower. And then the surface area of the membrane where diffusion is taking place. So if you have more surface area per volume versus less surface area, okay, per volume, then you're going to have a much more rapid rate of diffusion. Okay, and that's just simply because you have, again, more places for diffusion to occur through. So the higher the surface area per volume, the faster the rate of diffusion is going to be. And then, of course, the diffusion distance makes a difference. Now, this with our plasma membranes isn't really going to change too much, but if you're talking about like diffusion across your skin or something like that of substances, obviously areas where you have thick skin, your things are going to diffuse much more slowly than areas where you have, thinner layer of skin. So diffusion distance also has an impact. Obviously, the larger the diffusion distance, the slower the rate of diffusion is going to be. Okay, so when we talk again about diffusion through that plasma membrane, we already talked about the fact that, nonpolar hydrophobic lipid core of the plasma membrane is going to block a lot of things. And that's why we have that hydrophobic lipid core, is so that it can block things out. So again, remember that the things that like to pass through are small and also not very polar, okay? Larger molecules, and especially charged molecules, are going to need to be assisted by carrier molecules, especially proteins, like those transmembrane proteins that are going to form channels, okay? So what are some examples of things that can diffuse freely? And I realized that I kind of skipped through this slide. And then I doubled up. So I apologize. You can actually get rid of this slide in your lecture. It's basically the same thing, but I'm just going to go to it because it talks about some examples of things that can diffuse fairly easily across the membrane. And so one example is the exchange of oxygen and carbon dioxide between the lungs and the blood. So if you look in the lungs, our lungs are going to end up in these things called alveoli. And then there's going to be, blood vessels that are going to wrap around those alveoli. And so what's going to happen is as we take in a breath of air, right, our oxygen is going to go down its concentration gradient. And it can do that because, again, it's this small molecule, not charged. You're going to move right down its concentration gradient right across the membrane through simple diffusion. So oxygen will diffuse into our blood, and then carbon dioxide, which is in the high concentration here in our blood, is going to move out of our bloodstream and into the lungs. Okay? So those two gases are really important in terms of understanding diffusion, simple diffusion, because those gases are going to diffuse simply right across the capillary membrane and the alveoli membrane. The movement of substances across kidney tubules. We'll talk about, again, water diffusing and sodium and chloride diffusing, certain ions that will also make their way across. But our ions are going to need help through these little protein channels. We'll talk about that coming up here in just a minute. And then we have, again, diffusion of things like sodium and potassium through channels as well in the plasma membrane. Now, the reason that sodium and potassium need these channels is not because they're large, right? They're small, but they're very charged. And so they need those protein channels to help them get across. Let me back up here just a minute. And I just wanted to mention that there are going to be, when we talk about concentration gradients, there are going to be kind of two types of gradients across a membrane. One of them that we're going to talk about is actually the concentration gradient that we've been talking about of solutes, right, across the membrane. But we're also going to talk about these electrical gradients as well. So when we talk about things wanting to move down their concentration gradients, we can also get electrical gradients as well, where we have lots of positively charged particles on one side and lots of negatively charged molecules on the other side. And because opposites attract, anyway, I don't want to get too much into this right now, but this becomes important for when we talk about the nervous system, because we will talk about charged molecules wanting to move down a concentration gradient as they're attracted to the opposite charge. So I'm just, again, kind of bringing that up here because there are these things called electrochemical gradients, where, again, chemicals are moving not only down their concentration gradient, but also they're being attracted to opposite charges as the charges build up across the membranes. Okay, so let me go ahead and get back into, so easy for me to get off track here. Let me go ahead and get back to these different types of diffusion. So again, some molecules, especially again those that are fat soluble, non-polar, small molecules, just like oxygen, just like carbon dioxide, even water molecules will move right across this membrane through what we call simple diffusion. Okay, very simply moving across the membrane. They don't need any protein channels. Now, again, remember that water will still move across more slowly because it is partially charged. So it will move more rapidly across the membrane if it has a protein channel. And so we're going to talk about that now here in just a minute. So when we talk about diffusion, again, there are these different types of diffusion. And so we can have facilitated diffusion, which means that we need like a protein that will help facilitate the diffusion process. So in this case, there's two types of facilitated diffusion that we're going to talk about. There is carrier-mediated facilitated diffusion. And so all that is where we have this protein carrier here that's specific for one type of chemical. So it's going to bind a certain substrate, and then when it binds that substrate, it's going to undergo this kind of conformational change and then release it to the other side. So for example, glucose is transported across the membrane this way because glucose is a really large, bulky molecule. And so again, it'll have these protein carriers, and the way that it makes it across the membrane is by binding to one of these protein carriers and changing the shape and being released to the other side via this carrier-mediated facilitated diffusion. So again, it's just a protein carrier that's mediating that diffusion, facilitating that diffusion. Okay, if that helps you kind of remember it. Then we also have channel-mediated facilitated diffusion. Okay, so they're both facilitated diffusion, right? We've got the carrier-mediated and then the channel-mediated facilitated diffusion, but they're both facilitated by these proteins. This one just means that here it's through a protein channel. And so usually, again, this protein channel is polar, okay? So that way then it allows polar molecules to get through, or small lipid insoluble molecules. Again, they're going to be polar. So things like sodium and potassium that are charged, okay, are going to diffuse through this type of channel mediated diffusion. And then, of course, we have osmosis, okay? So osmosis is going to be just the diffusion of water. We're going to talk a lot more about the diffusion of water in just a minute. But remember that some water molecules will diffuse just straight across the membrane, but because they're partially charged, it'd be kind of slow. So we speed that up with what we call aquaporins. Aquaporins are these protein channels that are for water to move through, okay? These nice polar protein channels that allow water to move right through it. So let's get into osmosis, okay? Osmosis, again, hopefully this is mostly a review for you. I just liked this mug, osmosis, like awesome. So the diffusion of awesome across a semi-permeable membrane. Of course, the real osmosis is the diffusion of water across that semi-permeable membrane. And it's important in osmosis to know that we have to have a membrane that is blocking some of the solutes, okay? Because if it's not blocking the solutes and the solutes are allowed to go across, and the solutes will just diffuse, right, down their concentration gradient. And water, again, will diffuse as well, but it'll stay the same level, right? Because everything's just diffusing freely. But where osmosis comes into play is when you have a semi-permeable membrane that is blocking a solute, like in this example, sugar, okay? So if you put a really high concentration of sugar molecules on this side and a low concentration of sugar molecules on this side, okay? So what I'm going to do is I'm going to write out, okay, high, oops, let me go to my Bamboo over here, high sugar. So if, again, we have lots of sugar molecules, that's actually going to leave less room for water molecules, right? Per this amount of volume, okay? And I think sometimes this is the easy way to like write out what's going on with osmosis here. And so here on this side, you have fewer sugar molecules, and this leaves more room for water molecules, okay? And so in this case, remember the sugar cannot move through the selectively permeable membrane, okay? So here in this case, right here, we don't have the protein channels that are allowing it to diffuse down, okay? So the sugar is blocked from diffusing, which means only water is going to diffuse here. So what that means is water is going to diffuse down its concentration gradient from where it's in a high concentration over here down to where it's in a lower concentration here. And as it does that, it's going to push, right, the water column up here. So again, that movement of osmosis, that movement of water molecules is going to be down its concentration gradient, okay? From where water is in a high concentration, right? But the solute is going to be low, okay? So water moves by osmosis from areas of low solute, but high water, right? And we focus on water, to areas where water is low or the solute concentration is high, okay? So hopefully that helps. I always think it's easiest to remember osmosis by what's going on with water. If the solute is low, water is higher on this side. And if the solute is high, water is lower on this side, and water just simply is moving down its concentration gradient. And that way it keeps things very simple for you, okay? Now, when you talk about osmotic pressure is going to be really important to understand when it comes to the kidneys. So I want to mention it here, at least so that you've heard of it when you get to 202. Osmotic pressure is simply just the pressure that needs to be applied to a solution to prevent osmosis. So for example, here in this case, where we talked about, again, you can pretend these are sugar molecules that can't get across. And as the water molecules diffused across, okay, the osmotic pressure would be how much pressure do you have to push back on that water column to push the water back so that it's equal on both sides. That's the osmotic pressure. Or basically how much pressure is caused by that desire to suck water molecules over to this side, okay? So I think that's important to remember because we'll talk about that in the kidneys a lot, okay? The movement of water molecules, again, causes pressures. So there's hydrostatic pressure, which is simply just the pressure of water. I mean, you guys know hydrostatic pressure, like if you've ever had a hose and then you put a nozzle on it, right? You can create higher hydrostatic pressure by putting a narrow nozzle on it, right? So hydrostatic pressure is simply just the pressure of water inside something that's contained, right? Pushing on that. So either pushing out to get out of the hose, or pushing, we use it mostly inside the cells, pushing on the outside of the cell, right? The actual simple pressure of water inside that cell, the water molecules inside that cell. And then there's osmotic pressure, which is the tendency of water to want to move into the cell via osmosis, okay? And so we're going to talk about this in the capillaries. We're going to talk about this a lot in the nephrons, in the kidneys. So again, I'm not going to focus too much on it now, but I want to introduce it so that when you get to 202, it's not the first time that you're hearing this. So hydrostatic pressure likes to usually push water, okay? And osmotic pressure likes to suck water, oftentimes into the cell, okay? So we always like to say hydrostatic pressure pushes and osmotic pressure sucks. And this is kind of important because things like edema are actually caused by, a buildup of capillary hydrostatic pressure, right? And so we can end up where there ends up being like too much fluid inside the tissues. So that's actually what causes our edema when we get edema. So again, that's just I like to introduce that. We're focusing obviously more on osmotic pressure right now. And that is simply just the more solutes inside a cell, the higher the osmotic pressure is going to be, okay? The more it's going to want to suck that water in, okay? So really quick, I want to talk about tonicity before I end this little segment. I know that this gets to be a little bit challenging because students are always like, no, not tonicity. But really quickly, I want to tell you where the word, it's a silly story about where the word tonic comes from. So tonic, actually the initial medicinal kind of use of the word tonic came from the early 19th century in India and kind of the tropical coast of the, for the British. They had to deal with malaria. Malaria was a problem. And so they used this medicinal quinine and they would dilute it in water. But it tasted bitter. It tasted really bad. But that was what they needed to take to help prevent malaria in these tropical coasts. And so to get rid of that bitter taste, they would mix it with soda and sugar to kind of mask its bitter taste. And so whenever you think of the word tonic, you can think of kind of the mixing of solutes, right, to make a tonic. So, that's what they called the tonic. So you can think of when they mixed these solutes, especially sugar, right, in with this solution to help prevent malaria. And so when you think of tonic, think of solute, okay? And I'll tell you why this will help you in just a minute. So tonicity is just the effect that a solution has on its cell volume. Now, if something is isotonic, if you know that the word iso means same, same, okay? Isotonic just means that this is the same concentration of solutes inside the cell. So again, tonic meaning like your solutes. So same solutes inside the cell as outside the cell. And when we're talking about these solutions, we're talking about the solutions that our cells are bathed in. So an isotonic solution would mean that here you have, a beaker and you have some of your cells, okay? And you put them in a solution where there's the same number of solutes per volume outside the cell as inside the cell, okay? Now, if we have a hypotonic, what does hypo mean? Okay, if you know that hypo means low, right? The solute means, tonic means solute, right? So we have a low solute solution. If we put our cells, I'm going to draw, sorry, I'm drawing off here. I'm going to draw this solution here, and I'm going to draw the cell to be giant, just so you can see. But here in a hypotonic solution, you're going to put your cell in a situation where there is a lower concentration, lower solute concentration outside the cell than there is inside the cell, okay? So remember, if there is a lower solute concentration, I'm going to put S for solute, that means there's a higher water concentration, okay? And if there's a higher water concentration, where's water going to want to move? Down its concentration gradient, because right, there's higher solute concentration inside and a lower water concentration inside. So water is going to move down its concentration gradient right into that cell, okay? And this can be bad because the water you can have too much diffusion of water, and the water can actually build up inside that cell, causes too much hydrostatic pressure in there, and then literally the cell just bursts, okay? So hypotonic solutions are bad for our cells because they don't like to be in those hypotonic solutions, and it causes them to burst. Now, a hypertonic solution, I wish I had a bigger area for you. I'm actually going to just get rid of that for a second. Excuse me while I just draw on where I'm going to be in the slide. So a hypertonic solution, okay, remember hyper is going to be the opposite now. Hyper means higher, right? So if we have a higher solute concentration, here, outside the cell than we do inside the cell, okay? What that means is higher solute, lower water, okay? And now, this time we have a lower solute inside the cell and higher water, right? More water, and water is just going to diffuse right down that concentration gradient out of the cell, okay? And so when you put cells in a hypertonic solution, and you guys kind of intuitively know this, right? Like if you were to put something in salt, you know that it's going to draw the water out, okay? That's a hypertonic solution. And it will cause that cell to shrink up, okay? This is why, again, you don't want to take your freshwater fish and put it in salt water, right? Very, very bad to put it in that hypertonic situation. So again, this is just kind of what we just talked about. Anytime that you take a cell and you put it in a hypertonic situation. So let me go back. If you put it in an isotonic solution, the same amount of water molecules that are going inside the cell. Let me see if I can actually find a picture of this here. So here's my isotonic solution. And if we're talking about this isotonic solution, again, same amount of solutes inside the cell as are outside the cell, same concentration, right? And so the same amount of water molecules, oops, pen's messing up there. The same amount of water molecules they're still going to be moving, is what my big point is. Water molecules are always moving. It just means that you have an equal number of water molecules moving into the cell as you do out of the cell, right? Okay. Now, when we talk about a hypertonic solution here, okay, and I'll talk about crenation here in just a minute. With that hypertonic solution, remember that we've put it in a solution with lots of solute, high solute, and low water, okay? Low water. And if it's low water, then again, water is going to move down its concentration gradient from where it's in a higher concentration inside the cell to out of the cell. And so if you do this with your red blood cells, okay, if you were to take your red blood cells and put them in a hypertonic solution, they're actually going to undergo something called crenation. And what that means is literally the cells are going to shrink up and they're going to kind of form these little projections like this. And this is very bad for your red blood cells because they can't carry oxygen efficiently this way. And they'll eventually just break down. So that's bad. So you don't want to put your blood cells in a hypertonic situation, but you also don't want to put them in a hypotonic situation. And that's because, again, you're simply going to cause them to lyse. So remember, hypotonic situation means that you have a situation where you're putting your cells in a lower tonic solute situation. And then that means that leaves more room for water molecules, so higher H for H2O. And so then the water is going to want to move down its concentration gradient into the cell. So you're going to have more water moving into the cell than out of the cell is what they're drawing here, right? So here they're drawing equal amounts of water moving in and out. Here, more water is moving into the cell, and it'll eventually cause that red blood cell, it'll take in so much water, build up so much hydrostatic pressure that the plasma membrane can't hold it all, and it literally is going to lyse or break apart, okay? And we call that hemolysis, okay? So hint, make sure that you understand tonicity. This is really important. If you have to draw it out like I did, if you need more help with this, I am happy to set up some time with you to talk about this. But it's very important to understand what happens when you put your cells in situations of hyper and hypotonic. And so there are clinical applications to this, right? Like if your plasma membrane is severely damaged and stuff is allowed to diffuse freely in and out, this compromises all of the concentration gradients that you have set up that help you do muscle contraction and all kinds of things. So for example, burn patients oftentimes lose precious fluids this way. That's why they can oftentimes be dehydrated. And they lose proteins, ions, again, all kinds of issues that can happen with that broken down barrier. Also, when you talk about intravenous solutions, this is kind of interesting in terms of different tonicities that can be given. Isotonic solutions are going to be given if you need to give blood quickly, okay? If you have a person and they need a lot of blood fast, okay, you're going to give them an isotonic solution because you don't want to cause any type of osmotic imbalance, right? So you are going to give them an isotonic solution that has the same amount of solutes, right, as surrounding their cells. Now, hypertonic solutions sometimes are given to patients with edema, and that's to help pull water back into the blood. So again, if you're putting a lot of solutes into the blood, that'll help draw water out of the tissues. But again, you don't want to give too high of a hypertonic solution because then you'll cause your blood cells to undergo crenation. So again, you have to be, there's a very fine line in terms of how hypertonic you can give a solution. And then hypotonic solutions shouldn't be given at all because you just are in danger of lysing your red and white blood cells. So they just don't give those. So again, kind of fun to talk about the application of those. I'm going to go ahead and stop here. I know that was a lot. Please let me know if you have questions. I'm trying to make these shorter, but there's just so much material. And again, these first few lectures are probably going to be longer than what I would typically give in person. But what I want to do is create a set of lectures that if you need a little bit more review, that material is here. For those of you that are like, okay, I'm bored out of my mind, I know all this, please feel free to like fast forward through these lectures, because I know that a lot of this is a review. But we have, again, such a mixed group of people, and I'm trying to kind of cover all my bases. So I will come back the next time here and talk about active membrane transport with you.